Welcome to Saitechinfo channel, my dear students. In today's lecture, we will be discussing about enzymes, vitamins, nucleic acids. So this is the last part of our biomolecule lecture series, part 5, class 12, chemistry. Let us explore at the outset enzymes. Enzymes are biocatalysts. Enzymes are biocatalysts. What is the role of catalyst? It activates, it enhances the reaction. Similarly, like a chemical reagent, the biological samples can also encourage or enhance or affect the rate of the reaction. See, we have got some major functions and that are controlled by these enzymes, that is biocatalysts, like digestion of food, absorption of suitable molecules. So whatever the food particle digested, it should be converted into the suitable molecules so that it can be absorbed by the system. And finally, we produce the energy. So all these functions are controlled by these biocatalysts. They are called enzymes. Enzymes are globular proteins, means they have the globular spherical structures. These enzymes generally have the suffix ase. Ase is the enzyme that affects a specific sugar. In fact, these enzymes are very specific for a reaction, and it is very specific on the reaction site, that is called substrate. They are named based on their function or the reaction. For example, maltase that catalyzes maltose. That is, maltose is converted into glucose units. Oxireductase. So oxireductase, they are responsible for catalyzing the redox reactions. So redox reactions are nothing but the oxidation and the reduction, they occur together. So these oxireductase catalysts, that is the enzyme, they catalyze the redox reaction, that is oxidation of a particular reactant and reduction of another reactant. Similarly, invertase that inverts the optical rotation of sucrose, like D2 dextrorotatory to levorotatory, and that is why that enzyme is called invertase. So likewise, based on the specific reaction and the reaction site and their function, these enzymes are named. In fact, they are required only in very small quantities for the progress of the reaction. And like catalysts, they reduce the activation energy and thereby supports the reaction. Example, see, that is when we hydrolyze the sucrose in presence of acid, the required energy, activation energy is 6.22 kilojoule per mole. Whereas the activation energy is drastically reduced to 2.15 kilojoule per mole when they are hydrolyzed by using the enzyme, and that enzyme is sucrase. Vitamins. Actually, initially vitamins were thought of containing amino acids, that is the vital amino acids. But later we came to know that they need not contain amino acids. All vitamins need not contain amino acid. So that is why we dropped the vitamin, that is, we dropped E and call it vitamin. So we retain the name vitamin instead of vitamin, vitamin. They are also required in small quantity in our diet. See, their role is maybe, the amount may be very least, but their absence or deficiency causes specific diseases, and that's why they are called vitamin deficiency diseases. Most of them cannot be synthesized in our body, and that is why we need to depend on plants, because these plants can synthesize almost all types of vitamins, and that is why you are advised to take a lot of vegetables. Essential food factors. So there is no doubt about that. These are essential food factors. Besides your basic biomolecules like carbohydrate, fat, protein, etc. Bacteria of the gut can produce some vitamins required by us. Don't think that plants alone can synthesize. Bacteria, and that too which are living inside our body, can produce vitamins. All vitamins are generally available in our diet. Different vitamins belong to various chemical classes. So it would be very difficult to define them on the basis of the structure. Organic compounds in the diet in very small amount, it performs a specific, that is, biological functions for the optimum growth of and health of the body. So what are vitamins? Vitamins are organic compounds in the diet. They are required in small amounts to perform some specific biological functions for what? For optimum growth and to maintain health. So these are the 6 key points to define the vitamins. So what are vitamins? They are organic compounds. Diet, they are available in diet. They are in the diet in small amounts, very, very small amount, and perform specific biological functions, and then it is meant for optimum growth and health, the maintenance of health of the body. Then they are designated generally by alphabets like A, B, C, D, but further named as subgroups like B1, B2, B6, B12. So excess vitamins should also harmful. We cannot take excess of vitamins. Vitamin pills should not be taken without the advice of the doctor. Since vitamin is required, we cannot take any other food. So it should be taken in very small quantity as per the advice of the doctor. Then vitamins are classified into two groups depending on their solubility. They are fat soluble and water soluble. So fat soluble vitamins are not soluble in water. They are soluble in oils and fats. Example, vitamin A, D, E, K. Whereas they are stored in the liver and adipose. Adipose is a fat storing tissue. What about the water soluble vitamins? B group vitamins and vitamin C, they are soluble in water. They are not soluble in fats and oils, but must be supplied regularly because it may urinate it. It will be readily excreted in urine, so that loss should be compensated. So it cannot be stored except vitamin B12 in our body, and that is why vitamin C and B should be taken regularly. Then We have got different types of vitamins and their sources and what are the deficiency diseases. You have to buy out all these things. Let me consider the first one, vitamin A. So, vitamin A, the main sources are fish, liver oil, carrots, butter, milk. So, if the vitamin A is not sufficient, then its deficiency diseases are xerophthalmia, that is hardening of cornea of eye, and that will cause night blindness also. Vitamin B2, B1, that is called thiamine. It is present in yeast, milk, green vegetables, cereals. And what is the disease? Beriberi, that is the loss of appetite, and there won't be enough growth. It's called retarded growth. Then vitamin B2, also called riboflavin. It is available in milk, egg white, liver, kidney. Here, the deficiency disease is keratosis. It means fissuring at corner of mouth and lips. So you will have some cleavages here. So such kind of thing will be, your lips will be affected seriously. And it indicates some digestive disorder, then burning sensation of skin. So the best remedy is to top up with B2, that is riboflavin. Vitamin B6 is also known as pyridoxine, that is present in yeast, milk, egg, yolk, cereals, and grams. So convulsions are the diseases. Vitamin B12, it is present in meat, fish, egg, and curd. And the disease is pernicious anemia, that is, it is RBC, red blood corpuscle, will be deficient in hemoglobin. So it will result in anemic problem. Vitamin C, ascorbic acid. So citrus fruit, amla, green leafy vegetables. And the disease is bleeding gums, also called scurvy. Vitamin D, that is, it is present in sunlight, so exposure to sunlight, then taking fish and egg yolk will be good for having this vitamin D. Suppose if it is deficient, then you will be struggling from rickets, that is bone deformities in children, and osteomalacia, that is soft bones and joint pain in adults. Vitamin E, they are present in vegetable oils like wheat germ oil, sunflower oil. And what will happen if it is deficient? There will be an increased fragility of RBC and muscular weakness. Vitamin K, that is, it is present in green leafy vegetables, then it increases the blood clotting. Suppose if it is deficient, that is, if vitamin K is deficient, it will cause blood clotting. And then now let us discuss about the nucleic acids. Nucleic acids, they are we are very familiar with these nucleic acids, that is DNA and RNA. Deoxyribonucleic acid and a ribonucleic acid or ribonucleic acid. First of all, the transmission of inherent characters from grandparent to parent, parent to grandchildren, like one generation to another, is possible because of chromosomes. And this phenomenon is called heredity. Chromosomes are made up of essentially DNA and RNA. They have long chain polymers of nucleotides, and they can also be called polynucleotide. Now let us look into the chemical composition of DNA and RNA. Hydrolysis of DNA or RNA yields pentose sugar. So pentose sugar means you will have 5 carbon atom, a sugar with 5 carbon atom, pentose sugar. Phosphoric acid, that is H3PO4, phosphoric acid. And then nitrogen containing heterocyclic compounds. What is that? Nitrogen containing heterocyclic compounds. Now we have got various types of heterocyclic having nitrogen, like pyrrole, pyridine, etc. So DNA molecules, the sugar moiety means part. The sugar part is beta D 2-deoxyribose, but in RNA it is beta D ribose. So here in DNA you have this sugar part. So DNA molecule contains this sugar. And what about this? This is present in RNA. Okay, so this is a sugar molecule. You can count, actually there will be 5 carbon atom, 1, 2, 3, 4, 5. So we have identified this structure as a furanose, we already know. In the case of sucrose, fructose we have studied. So similar to that. So beta 2, so beta 2 deoxyribose means that is you have OH. You just compare the structures here, OH, H, and what happened here? There is no oxygen, and that is why it's called deoxy. Whereas here you have the oxygen, so that is ribose. So since it is it doesn't have oxygen, it is called a deoxyribose. You should identify here. Okay, whereas you don't have oxygen here, and that is why it's called deoxy. Are you able to understand this? Right. Then, and particularly why it's called a D2 dextrose structure, then dextrose branch, and 2 represents in the second position, the oxygen is left here. Do you understand this point? Right. Then DNA contains basically 4 bases. As I already discussed, the bases are nothing but the heterocyclic compounds having the nitrogen. So they have the 4 bases. Heterocyclic means what? They have got the cyclic structure, but apart from carbon, there will be heteroatoms like nitrogen or oxygen. So in this case, nitrogen. So you can see here, adenine is one of the structures. So this is adenine. So DNA contains 4 important bases, adenine, and this is called guanine. So all are heterocyclic with carbon-nitrogen arrangements. And this is cytosine, and this is thymine. I have highlighted here with the yellow because in RNA also you have all these 3, but the 4th will be different. Okay, so let us look at. So here all are same in RNA, but instead of that is thymine, you have here uracil. Are you able to follow? So that is different. So that makes the difference between DNA and RNA. And then let us talk about the structure of nucleic acids. We can have nucleoside and nucleotide. Let us discuss what is nucleoside. So the nucleoside will have this structure, whereas nucleotide will have this structure. So what makes the difference between these two? Can you find out the difference? Here there is no phosphate unit, whereas in the nucleotide you have the phosphate unit. Phosphate means PO4 what? 3 minus. So you've got that phosphate unit. So a unit formed by the attachment of one position of sugar. So this is the one position, one dash position of sugar, and the sugar carbons are numbered 1 prime, 2, 3, 4, 5, etc. in order to distinguish from the base, because in the base also you have got carbon. So in order to distinguish, we have put 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Okay. Whereas nucleoside linked to phosphoric acid can be called nucleotide. So what is nucleotide? It is a nucleoside with the phosphate unit. Do you understand this? Right. Then, now, structure of nucleic acids. So the nucleotides, they combine together to form phosphodiester linkage. What is that? Phosphodiester linkage. So this is the phospho, okay, and diester linkage, right? So phosphodiester linkage. See, now you have got one peptide, that is with phosphate unit, sugar unit, base unit. As we already know, the nucleic acid contains these three important components. What are those? Phosphate, sugar, base. Now let me combine these two, so that on one side, that is at the fifth carbon atom, there is a phosphate link. Is it not? So, and that, I mean, through the third carbon atom, it is linked. So this 3 and OH, are you able to understand this? That is, this OH and O is joined. These two are joined, so that, and this is called, actually here, it joins here. Okay. So it forms phosphodiester. Are able to follow? It's called phosphodiester linkage. Okay. So that results dinucleotide. So likewise you have got that is polynucleotide. Okay. Right. Now let us have this nucleic acid. The primary structure of nucleic acid. It gives the essential information regarding the sequence of nucleotides in the chain of nucleic acid. As we already know, you got the sugar, base, phosphate, and through phosphate there is a link. Now sugar, base, phosphate. So N times. So likewise it goes on, and that's why we call it polypeptide. I mean poly. So here, look at this. Polynucleotide, not peptide, polynucleotide. Right. Nucleic acids have secondary structure also. James Watson and Francis Crick, they gave a double strand helix structure. So this is the double strand helix structure. And they are bound by the hydrogen bonds. They are bound by the hydrogen bond between the pair of what? The bases. The two strands are complementary to each other because these are bound by the hydrogen bonds between the specific pairs of bases. You could have seen adenine, guanine, thymine, so such a combinations are there. Then, and adenine forms a hydrogen bonds with the thymine, and cytosine forms a hydrogen bond with the guanine, and they are here simply represented as that is A and T. So adenine, this is A, and T, thymine. Are you able to understand? Similarly, guanine and cysteine. Okay, right, cytosine. Then in secondary structure of RNA, so we were discussing about the DNA. Now RNA, there are also helices present. They are present, but only single stranded. Excuse me. Sometimes they fold back themselves to form double helix also. So RNA molecules are of three types. They perform different functions. What are those? That is messenger RNA, ribosomal RNA, then transfer RNA. So these are the different types of RNA. Now there is a very important topic, that is the DNA fingerprinting. every individual has got some unique fingerprints. We have got some unique fingerprints, particularly index finger or thumb we have. But that can be altered by some surgical operations, is it not? So we want to have this identity in a better way, in an advanced manner. So for which the DNA fingerprinting is useful, because the sequence of bases on DNA is very, very unique to every individual. So information regarding this called fingerprinting of DNA, that is DNA fingerprinting. It is the same way for, in fact, every cell has got the same pattern and cannot be altered by any known treatment. And that is why DNA fingerprint is nowadays used in forensic laboratories to identify the criminals. Then to determine the paternity of an individual. So we can find out who is the father. Then to identify the dead bodies at any incident by comparing the DNA of parents and children. And then it is also used to identify the racial groups and to rewrite the biological evolution. So these are all the features of DNA printing. And finally, we need to discuss about the biological functions of nucleic acids. So DNA is the chemical basis of heredity. They may be regarded as the reserve for genetic information. So DNA is exclusively responsible for maintaining the identity of different species of organisms over millions of years. We can have the DNA bank. So a DNA molecule is capable of self-duplication during the cell division. So identical DNA strands, they are transferred to the daughter cells. And then protein synthesis is a very important one, and it's a very important function of nucleic acids. It is done by various RNA molecules. And in fact, this is the message for the synthesis of a particular protein that is present in DNA. So I hope you should have enjoyed this video. So in biomolecules, carbohydrates, proteins, fats, vitamins, minerals, these are all very, very essential ingredients for every human being or every creature. So after all, they are basically required, and it is a very interesting thing to study such essential food items, essential items which are present in the foodstuff. Okay, so thank you very much, and please go through this lecture. Just play and pause the button whenever you want to have further clarification, and always take down notes when you watch any educational video. Thank you very much for watching this video.